Good morning. Thank you for joining us today for our Farm Foundation Forum, Antimicrobial Stewardship in Agriculture. How far have we come and what's next? We are glad to have this opportunity to engage with you today and are thankful to Farm Credit for their support of this forum. My name is Martha King, and I am the Vice President of Programs and Projects for Farm Foundation, located just outside of Chicago, Illinois. I'm looking forward to this morning's discussion on antimicrobial stewardship in agriculture today. Before we get into today's program, I'm going to take just a few moments to share a bit more about Farm Foundation. We are a 501c3 nonprofit working at the intersection of agriculture and society to address challenges that affect the entire food and ag value chain. Specifically, we are an accelerator of practical solutions for agriculture, accelerating people and ideas into action. The three levers we use to accomplish this are policy, innovation, and education. Forums such as today's are just one part of our extensive program of work, which is guided by our mission to build trust and understanding at the intersection of agriculture and society, and our vision to build a future for farmers, our communities, and our world. We rely on partnerships to fund our work and increase our impact. So if you're interested in learning more about funding or partnering with us, I invite you to reach out to explore collaboration. Now, I'd like to take a minute to highlight our Friends of Farm Foundation program. With your enrollment as a friend, you will not only be helping to support the mission and vision of Farm Foundation, but you will also gain exclusive benefits such as first reads of our issue reports, networking opportunities, and much more. Being a friend is an investment in building a better future for farmers, our communities, and our world. To learn more about becoming a friend of Farm Foundation, go to farmfoundation.org/friends. The link is also being posted in the chat. In addition to learning more about Farm Foundation and our work by visiting our website, I encourage you to connect with us on our social media platforms. If you are posting on social media about this morning's session, we ask that you please use #FarmFoundationForum. And here are our last few housekeeping notes. There will be an audience question and answer session at the end. We'll be using the Q&A button at the bottom of your screen, not the chat, please, to queue questions. This forum is being recorded and will be posted on our website at farmfoundation.org, as well as on our YouTube channel. We'll send out that link following today's program. When the forum concludes, you will receive a link to a brief survey. We appreciate your feedback and your time in completing the survey. Now let's turn to today's forum topic. We have an outstanding panel of experts with a diversity of expertise on animal and crop health and antimicrobial resistance. In addition to discussing regulatory changes affecting antibiotic use in livestock production, the conversation will also be broadened to consider proper stewardship of antifungals in crop production. In recent years, Farm Foundation conducted a number of workshops around the country and hosted a major summit in 2016 related to FDA guidance and the Veterinary Feed Directive, resulting in a comprehensive report on the stewardship of antimicrobial drug use in food-producing animals. You can learn more about that report and effort on the Farm Foundation website. It is now my pleasure to introduce our moderator and discussion leader, Dr. Paul Plummer. Dr. Plummer is Executive Director of the National Institute of Antimicrobial Resistance Research and Education, as well as a professor and Anderson Chair in Veterinary Science in the Department of Veterinary Diagnostic and Production Animal Medicine at Iowa State University. He also serves as the Associate Dean for Research and Graduate Studies at the Iowa State University's College of Veterinary Medicine. His academic role as both a board-certified food, animal, internal medicine, and infectious disease specialist, coupled with a doctorate in veterinary microbiology and his leadership of an active research lab, placed him at the intersection of translational research focused on AMR. In addition, he serves as a voting member of the Presidential Advisory Council for Combating Antimicrobial Resistant Bacteria and serves on the American Veterinary Medical Association Committee on Antimicrobials. Paul, thank you for moderating our forum today.
Thank you, Martha, for the opportunity and the privilege of moderating this excellent panel today. I'm looking forward to the discussion, and as we get ready to launch here, I just wanted to provide a few comments about NIAMRE, or the National Institute for Antimicrobial Resistance Research and Education, where I serve as an executive director. Our goal is coordinating action to combat the global threat of antimicrobial resistance. And if you go to the next slide, you can see our vision and our mission described there. Our vision is certainly to be a trusted leader in coordinating One Health efforts that preserve the ability to prevent and treat infectious diseases for generations to come. And we use a One Health approach, focusing on optimizing simultaneously the health of humans, animals, and the environment to work towards the mission of driving cross-sector engagement and coordinated action to combat the global threat of antimicrobial resistance across all One Health sectors. The next slide very briefly demonstrates or shows some of our current members that participate in NIAMRI events. And so we have memberships for both industry, academic, as well as affiliate groups. For more information about membership, you can see Farm Foundation is a member. On the next slide, you can visit us at niamri.org. Of interest, we have particular projects going on right now related to interprofessional education and growing education across the One Health sectors, having physicians, veterinarians, crop scientists, and extension agents, and a variety of other specialties working together to talk through antimicrobial resistance issues. We also partner with a number of federal agencies on cooperative agreements related to antimicrobial resistance in the One Health space. And we have several upcoming events as well that I would encourage you to go visit us at our website to learn more about. With that background on NIAMRI, we're now going to transition into our presentations from each of our panelists. We're first going to hear from Dr. William Flynn, Deputy Director of Science Policy at the Food and Drug Administration. Bill is a veterinarian and graduate of the Ohio State University, and he joined FDA's Center for Veterinary Medicine in 1993 and has been CVM's Deputy Director for Science Policy since 2011. Welcome, Bill, and we look forward to your comments.
Good morning. Thank you, Paul. And thank you for the Farm Foundation for hosting today's forum and for the opportunity to participate. I did want to spend a few minutes just reflecting a bit on some of the actions that FDA Center for Veterinary Medicine has taken over the past 10 years or so, provide some updates on some ongoing initiatives. Reflect a bit on sort of data and what that information is, what that data is showing us in terms of impacts on resistance and antimicrobial use. And then finally sort of touch on what we see as a next step and sort of our vision for moving forward from here. Next slide, please. So FDA, our focus really has been on the concept of antimicrobial stewardship and looking for ways we can help support optimizing how antimicrobials are being used in the animal agricultural sector to manage disease, and at the same time focusing on what we can do in terms of addressing the broader public health concerns about antimicrobial resistance. We've kind of been focusing on sort of three areas. One, looking at the products themselves as FDA is the regulatory agency responsible for approving drugs that are used in food producing animals, and that includes antimicrobials. We've also focused on what we can do in terms of supporting stewardship at the level of the user with veterinarians and at the level of farms. And then also it's important that all these activities are guided by science and what that science is telling us. And so it's really important that we emphasize and ensure that we have the data we need to monitor antimicrobial use and resistance. From a approach or strategy standpoint, again, we've really focused on stewardship and looking at it from a risk-based standpoint, focusing on those drugs that we believe pose the greatest risk, and those are the drugs that we've referred to as medically important antibiotics, and these are the drugs that not only do we rely on them for treating disease in animals, but we also rely on some of the same drugs or drugs in the same or similar classes of drugs for treating disease in human health. And so it's important that we try to do what we can to make sure that we're using them as judiciously in the food animal sector to minimize contribution to antimicrobial resistance. And then from an approach standpoint, we've been emphasizing a collaborative approach to bring all the affected stakeholders to the table to try to help support strategies moving forward. Next slide, please. And just reflecting back over the past 10 years, a couple of key, milestones. Going back to 2013, we initiated a strategy to work collaboratively with animal pharmaceutical companies to phase out the use of growth promotion, use of medically important antibiotics used in feed or drinking water, and shift the use of those products from over-the-counter status to status that requires veterinary oversight. That important initiative was successfully fully implemented with those changes going into effect in 2017. In 2016, recognizing the need to have better data to sort of help inform our activities, we did initiate some studies or funding to support studies with researchers both at Kansas State and University of Minnesota, trying to develop better strategies for how we can collect data on antimicrobial use to help inform antimicrobial stewardship efforts. In 2018, we published a 5-year antimicrobial resistance plan, which has been guiding our efforts since that time. And I'll touch more a little bit on that at the end. In 2020, the Noms program, which is the National Antimicrobial Resistance Monitoring Program, published its strategic plan. A key sort of theme of that plan is sort of focusing the efforts of the Noms program, which is focused on monitoring resistance in the food animal sector, focusing those efforts more on a One Health approach and emphasizing the importance of One Health. In 2021, we initiated a strategy to look at any remaining medically important antimicrobials that still are available over the counter and put in place strategies for transitioning those remaining products. So while the feed and water products were transitioned to vet oversight in 2017, there still remain certain products, a more limited number, but certain products that we recognize that some folks do rely on accessing these products that are still available over the counter. But again, recognizing the importance of having veterinarians involved in the use of antimicrobials, we have initiated a plan to transition those remaining products to veterinary oversight as well. Next slide, please. And so just touching on a couple ongoing initiatives, as I mentioned, an important aspect of what we've been doing in terms of our responsibility for ensuring safety of drugs, including antimicrobials. For almost 20 years now, we've had a process in place, which is a risk assessment process for evaluating antimicrobial risk for any new products that are being developed for use in food-producing animals to assess the potential risks they may pose to contributing to resistance development, and taking that into account as those products are developed and put into the marketplace. A key aspect of that, again, is taking into account the relative importance of that particular drug to use in human medicine. And again, this is sort of keying in on the One Health aspect of this issue. Importance of this issue is on looking at it from that standpoint, given that there is sort of this crossover between relying on the use of these products, both for use in animal medicine, but also for human medicine. And so we are in the process now of revisiting that process for ranking drugs with regard to their human medicine, to take into account any changes in practice of medicine that have occurred over almost the last 20 years since we initiated that process. Next slide, please. As I mentioned, while we shifted all medically important feed and water uses of antimicrobials to veterinary oversight in 2017, there still remain some products available over the counter, including some injectable products and some other dosage form products that also contain medically important antimicrobials. Again, recognizing the important role that veterinarians have in helping to ensure and optimize stewardship and optimizing the use of these products. We have initiated a process in 2021, which lays out essentially a 2-year phase-in period. So our target date is June of 2023. We do expect at that time that the products that are over the counter now will begin to shift over to prescription status, at that time. And this is an effort that we've been working with the pharmaceutical companies who have been collaborating with us to put this plan in place and to implement it by June of 2023. We're working now to help communicate or put information out to those that may be affected here in the lead up to that June date. We certainly recognize that farmers and ranchers, and particularly in certain parts of the country, have relied on accessing some certain products from various farm supply and other sources. And certainly it's important that they continue to have access to the products they need and want to make sure they understand what sort of changes are coming and emphasize the need to start planning for that and working with their veterinarian or making contact with a veterinarian that can help make sure that they have the products they need on hand when these changes go into effect, and that they can have those products on hand when they need them in the future. Next slide, please. Another issue related to, again, with the idea of us looking at ways we can help to mitigate or slow down the rate at which resistance may develop in conjunction with the use of antimicrobials in food animals is looking for ways we, again, optimize how these products are being used. One issue relates to the use of these products as medicated feeds, and better looking at ways that we can better target when and for how long these products are being administered. Again, these products are now under the oversight of veterinarians, and we're looking to update the labels of certain of these products that currently do not have defined instructions on their labels as far as duration of administration. We believe it's important that, again, we strive to target, the use of these as best as possible to treat, control, or prevent disease, but minimize the extent of use in order to slow down resistance. So this is something, again, we're working on with the pharmaceutical industry to again, refine how these products are labeled to better target and optimize how they're being used. And so this is an ongoing initiative. We expect more. We published the concept paper last year to get some initial input. We expect to be developing another document as a draft guidance document. We'll tend to seek additional public comment on that next year. Next slide, please. As I mentioned, I think an important aspect of us addressing AMR is having the information and data we need to assess whether the actions we're taking are having an impact and be able to adjust our efforts in response. Sort of three areas we've been focusing on is collecting information or data on antimicrobial resistance itself and monitoring trends. As example is the Noms program, collecting and reporting data on antimicrobial sales. That's the volume of amount of product that pharmaceutical companies essentially put into being used at the farm level. Next slide, please. Next slide, please. There we go. And so the first, looking at sales data, I think it's this slide just provides an overview of the past 10 years, essentially, or from 2011 to 2020, looking at sales data. And this is data that drug sponsors companies report to FDA, and we publish these annual summaries. What's notable here is just reflecting on sort of what we've seen over time. And so here are these bar graphs that show total volume of antimicrobials sold or distributed each year that we consider medically important antimicrobials. And just looking at the trend here, the blue line down the middle is sort of meant to sort of represent, a key timeline or time point or milestone when, again, we made the changes to shift products to veterinary oversight, the feed and water products to veterinary oversight and eliminate growth promotion. And it's notable that we saw a pretty substantial change in shift downward in terms of volume of antimicrobials sold. Remember we initiated that strategy in 2013, and that fully went into effect in 2017. And between the high point of sales, which was 2015, we've seen about a 38% decline in overall volume of sales, and that essentially has that lower level now has remained pretty consistent since 2017. Next slide, please. And looking at resistance, I know this is a busy slide, but I just wanted to provide this as sort of an example. Again, this is looking at E. coli from on the left, turkeys and chickens, and then on the right, the graphs are beef cattle and swine. The colored lines going from left to right are trends in resistance for different drugs. And that purple, I guess, is purple line going vertically, again, sort of indicating that sort of 2017 milestone time point. The only point I'm making here is just looking at overall trends. And I think it's a bit of a mixed bag in terms of what we're seeing, but overall trends are either sort of holding steady in terms of where they've been, or we have seen some declines, which is certainly encouraging. Next slide, please. With regard to actual antimicrobial use data, we currently have very limited sort of infrastructure in place for capturing and tracking this kind of information. As I mentioned earlier, we have been supporting some studies to sort of develop methodologies for collecting this kind of information. Again, initiated funding for studies in the food producing animal sectors, cattle, swine, turkeys, and chickens back in 2016. Those projects, which went for 5 years or so, are wrapping up now. We also initiated some similar studies in the companion animal sector for dogs and cats in 2020, and those are ongoing now, because clearly this is an issue that's not just limited to the food animal sector. It's also, we have to look more holistically at other places where antimicrobials are being used, including in our pets. And those proposals, I think have gotten some valuable information from those, as those studies have sort of developed, examined different methodologies for how to capture this kind of information. And next slide, please. So when you're looking at data, I talked briefly about sales data, and it does have some limitations, because sales information is simply representing what is going into distribution channels for sale. And it doesn't necessarily reflect what is actually used at the farm level, and it doesn't really give us any specific information about how that drug is being used. And I think that's an important point, because again, our focus is really on stewardship and how these drugs are being used in terms of optimizing how they're being used, and identifying things that we can do to better support stewardship and use these drugs as a smart way as we can, right, in terms of addressing the animal health needs that exist, but at the same time minimizing the impact on resistance. So with that in mind, we are looking and have been working on a project now, looking at ways we can put in place infrastructure to better capture or collect actual antimicrobial use data. We have an ongoing project with an organization called the Reagan-Udall Foundation for FDA, who's been helping to facilitate some stakeholder engagement to gather information on how we might, on different models for potentially some form of public-private partnership model for collecting this information. And again, we think there's real benefits to having access to this kind of information for helping to monitor trends and inform what's happening in terms of AMR, and help gauge or guide us in terms of developing and implementing stewardship activities at the level of farms. Next slide, please. And then in terms of what's next, I think as I've already sort of outlined, we are in the sort of latter stages of our 5-year plan. we're working to advance a number of the initiatives within that plan, and including the transition of products to prescription status in June 23, refining how we determine or define what drugs are considered medically important and how important those drugs are for human medicine. That helps to gauge how we help to mitigate or address AMR risks with those use of those drugs in animals. Better defining the use duration issue for feed use products, turning more attention towards the companion animal sector and what do we need to be doing there in terms of how we're using these products in our pets, as well as looking at what are the data gaps and what information do we need to better guide our activities with regard to addressing AMR. And again, as we approach the end of our current 5-year antimicrobial resistance plan in 2023, we're now in the process of sort of looking at and developing our strategies for what's over the horizon in terms of what additional efforts are needed to address AMR. Next slide, please. And in conclusion, again, I think we have made some important progress forward, and important changes have occurred in terms of addressing AMR. We do think it's important to continue to focus our efforts on stewardship and what can we do to optimize how we're using these products to address animal health issues. And then last, from an approach standpoint, we think it's critical that we approach this by using a collaborative approach and bringing all stakeholders to the table. Again, thank you very much for the opportunity to participate in today's forum.
All right. Thank you, Bill. We appreciate those comments, and we'll look forward to having you engage with questions here after our additional speakers. Now we're joined by Dr. Chelsea Shively. She's currently the Antimicrobial Resistance Coordinator in the Office of Interagency Coordination from USDA APHIS Veterinary Services. Prior to this, Chelsea spent 3 years working in the National Animal Health Monitoring System team focused on collecting data on antimicrobial use and resistance in animal agriculture. She served as an AVMA AAAS Congressional Science Fellow in 2016 to 2017, working on the U.S. Senate Committee on Agriculture, Nutrition, and Forest, and is also board certified by the American College of Animal Welfare. Thank you for joining us, Chelsea, and we look forward to your comments from an APHIS perspective.
Thank you, Dr. Plummer, and I want to say I really appreciate the invitation by the Farm Foundation to participate in this forum today. I will be sharing some of the activities in APHIS related to antimicrobial use and resistance in animals. Next slide, please. So when thinking about how far we have come on addressing AMR, the story for APHIS really begins back in 2015 when the US government published the first National Action Plan for Combating Antibiotic-Resistant Bacteria, or CARB, providing a roadmap for the federal agencies to summarize their activities on this issue. The USDA then developed the USDA Antimicrobial Resistance Action Plan. USDA possesses in-depth knowledge of the management practices and technologies associated with animal health, welfare, productivity, and food safety. As such, USDA is uniquely positioned to contribute to the body of scientific knowledge about antimicrobial resistance, and specifically about the role of antimicrobial use in animals. The US government published the second CARB National Action Plan in October 2020, articulating actions to address AMR for 2020 through 2025. Next slide, please. So zooming into the role that veterinary services plays within APHIS, there's 4 key areas that we use to address antimicrobial resistance. The first is antimicrobial use and stewardship monitoring. This primarily is done through surveys conducted by the National Animal Health Monitoring System, or Noms. Through its representative study design, Noms provides national estimates of antimicrobial use, as well as important context about the overall animal health, including diseases that lead producers to need to use antimicrobials. Noms also collects data on antimicrobial stewardship practices, such as the use of a veterinarian, record keeping, and participation in training programs. Second, we do antimicrobial resistance monitoring. The Noms program collects antimicrobial resistance data from our healthy animal population. And then we also have the National Animal Health Laboratory Network that has a pilot project looking at antimicrobial resistance in animal pathogens in cattle, pigs, poultry, horses, cats, and dogs. Third, we conduct education and outreach. This is primarily through the National Veterinary Accreditation Program, in which we have freely available online modules to help educate veterinarians about antimicrobial stewardship. And finally, we conduct a lot of collaborative projects with industry and university partners. These projects help us to better understand antimicrobial resistance in animals. We're partnering with veterinarians to look at trends in use and resistance, and working with academic researchers to improve sampling for antimicrobial resistance on farm. Next slide, please. So I want to talk a little bit about the National Animal Health Monitoring System, as that's where a lot of the work that we do related to antimicrobial use and resistance occurs. So the Noms program provides information about antimicrobial resistance in our healthy animal population. The unique study design of Noms studies allows us to provide national level estimates. While collecting data on antimicrobial use is important, it is also important to understand the stewardship activities that producers are using, such as record-keeping practices, using veterinarians, and participation in training programs. It is also important to understand antimicrobial use in the context of overall animal health, including the diseases that lead producers to need to use antimicrobials. Antimicrobials are one tool of many that producers use to address animal health challenges on their operations. Next slide, please. I want to highlight a couple of recent Noms studies. So in 2021, we conducted the swine study. This included both large and small enterprises. And from that study, we will have updated antimicrobial use and stewardship information, as well as antimicrobial susceptibility testing for Salmonella, E. coli, Campylobacter, and Enterococcus. Also in 2021, we conducted a health management on U.S. feedlot studies, where we will have updated information on cattle health practices, including antimicrobial use and stewardship. So this study was survey-based only, so no antimicrobial susceptibility testing information was collected. These two studies will provide what we are saying the after. So we conducted similar surveys back in 2017, asking about producer practices on farm in 2016 before implementation of the veterinary feed directive changes, as mentioned by Dr. Flynn. So now in 2021, we will be asking producers what they're doing now and try to understand how the FDA policy changes impacted producers and their practices on farm. We're finishing up reporting on our Noms cow-calf 2017 study, where again, we have updated antimicrobial use data as well as susceptibility testing. And also working on our goat 2019 study, where we have updated antimicrobial use data and some susceptibility testing information. And notably, the goat study was the first time that we partnered with one of our states. We worked with the California Department of Food and Agriculture on their antimicrobial use and stewardship program to sample goat operations in California in order to help meet their information needs on antimicrobial use in the state. In addition to collecting information on use and resistance, Noms studies collects data on a variety of topics, including animal health, management, and biosecurity. This provides additional context for interpreting antimicrobial use data. Some upcoming Noms studies include bison, sheep, aquaculture, and backyard animal keeping. So more to come. Next slide, please. So I want to talk now about the National Animal Health Laboratory Network AMR pilot project. This project is now in its 5th year, and it is focused on developing a surveillance stream to monitor antimicrobial resistance profiles in animal pathogens routinely isolated by veterinary clinics and diagnostic laboratories across the U.S. And it's really our first look at antimicrobial resistance in animal pathogens in our sick animal populations. This project covers 4 livestock species, cattle, swine, poultry, and horses, as well as 2 companion animal species, dogs and cats. Reports summarizing the findings are published annually, showing trends in antimicrobial resistance across livestock and companion animals. This project was originally designed to be a 5-year pilot project, and it is currently transitioning into a long-term monitoring program. This project recently published an online dashboard to display the results of antimicrobial resistance in near real time. A sample screenshot is shown here. The dashboard is interactive, allowing the viewer to select the animal species, the bacterial species, specific resistance profiles, as well as the date range that they are interested in. Having a better understanding of antimicrobial resistance in animal health pathogens will help veterinarians and producers make better treatment decisions and hopefully reduce the use of ineffective antimicrobials. Next slide, please. Optimizing antimicrobial use will require education and training for all users of antimicrobials, including producers and veterinarians. The National Veterinary Accreditation Program provides accredited veterinarians with training, and we have 2 modules specifically on antimicrobial stewardship and judicious use, as well as 1 module on honeybees, which must now have veterinary involvement for obtaining antibiotics through the Veterinary Feed Directive. We will be updating this training based on upcoming FDA policy changes mentioned by Dr. Flynn, such as moving the remaining over-the-counter antimicrobials to prescription status. Next slide, please. I want to take this opportunity to highlight a few of the collaborative projects that we have among many. So first, I want to talk about this relatively new collaboration with a private veterinary clinic in Minnesota that has been collecting antimicrobial use and resistance data of some of their swine clients. This collaboration will provide data looking at antimicrobial use and resistance on commercial swine operations and includes resistance in both human and swine pathogens. Data collected through this collaboration will allow us to look at trends in resistance, as well as better understand why certain swine operations use more antimicrobials than others. So far, this collaboration has been quite successful, and using trusted veterinarians has helped gain support with producers for sharing these sensitive data. Next slide, please. Another new collaboration that we are just getting started with now is with the dairy industry in New York State. This project will hopefully identify gaps in knowledge as well as structural and social barriers to on-farm change. Anecdotally, most dairy producers, when asked, are already using antimicrobials judiciously, so it can be difficult to get producers to change practices based on the latest research. For example, many dairy farms could benefit from changing their dry-off procedures to using a selective dry cow therapy program. Yet many producers are resistant to changing their practices. We're hoping that this project will help us to better understand some of those barriers to change on farm. And we're looking at the problem from a different approach by talking about disease reduction rather than specifically talking about antimicrobial stewardship. And we're hoping that that approach will improve our engagement with dairy producers. Next slide, please. I've given a brief overview of what we do in APHIS to address antimicrobial resistance, so I think it's important to mention how we are able to do this work. APHIS relies entirely on voluntary participation of producers, veterinarians, and diagnostic laboratories to work to address antimicrobial resistance. This requires trust, and an important component of our work is protecting confidentiality. APHIS also collaborates across the federal government through programs such as Noms, as well as with state, university, and industry partners. APHIS does not have the resources, the reach, or the regulatory authority to address antimicrobial resistance alone. Next slide. It is important that we communicate the work that we are doing to address antimicrobial resistance to stakeholders that can help implement solutions. Historically, many of our programs related to antimicrobial resistance have published lengthy PDF reports like the one shown here. These reports are utilized by many academic researchers but fail to reach producers. APHIS is now developing interactive dashboards that allow near real-time reporting of our results, such as this dashboard shown here from the Noms AMR pilot project. Improving our communication tools helps us to reach new audiences, such as getting key information back into the hands of producers about antimicrobial use, since they are the ones with the ability to optimize use on the farm. These new communication tools will also hopefully help us to be more timely with our reporting. Next slide, please. For all of the APHIS activities to address AMR, it is important that we use a science-based approach. The monitoring and surveillance activities we have help to identify trends. Research helps us to understand the cause and impact of these trends. And education and outreach helps us convey information and feedback into the surveillance and research activities to help us to continually improve our scientific understanding to target resistance in collaboration with our USDA, other federal and external partners. Next slide, please. When thinking about where we are going on antimicrobial stewardship, in APHIS, we continue to explore new data sources and partnerships on antimicrobial use and resistance. APHIS received new funding in FY 2021 to develop an antimicrobial resistance dashboard through a public-private partnership. We have established a cooperative agreement with NIAMRI and Dr. Plummer to evaluate options for data protection and security, a necessary first step before developing a dashboard. And planning is ongoing on how to best utilize this funding to support ongoing AMR activities. We also recently hosted a department-wide virtual public meeting on antimicrobial resistance, and that included an opportunity for public comment. USDA will be revisiting the USDA AMR action plan over the next year or so, identifying new opportunities to address AMR. And also, APHIS continues to engage with other partners, such as FDA, and looking at the future of antimicrobial use data collection on farm. Next slide, please. We can all agree that antimicrobial resistance is a One Health challenge, highlighting the interconnectedness of humans, animals, and our shared environment. Historically, we have focused on livestock as a source of resistant infections in humans, primarily through food. As we look to the future, APHIS has been expanding the scope of One Health to also include wildlife and companion animals. When looking at wildlife, our National Wildlife Research Center has been developing AMR bacteria diagnostics for wildlife and conducting experiments and field studies involving a number of species to determine their potential for transmitting disease to livestock and crops. Our NWRC has identified multiple wildlife species, such as raccoons, gulls, and European starlings, as hosts that can move AMR bacteria across wide areas, and in some cases, even continents. They have also found that feral swine samples collected at landfills show resistance to multiple antibiotics. In our companion animal space, Animal Care is developing new educational materials for dog breeders regulated under the Animal Welfare Act about antimicrobial resistance and the importance of antimicrobial stewardship. And that came out of an effort and a push from our colleagues at CDC, and you might hear a bit more about that from Dr. Nichols. And then finally, crops. APHIS has supported research on how antimicrobials are being used in crops, such as addressing citrus green disease or fire blight in apples and pears. And we hope to continue to expand our work in this area. Next slide, please. So again, I thank you for this opportunity to share information about what APHIS is doing now and with a look to the future about what we might be doing on antimicrobial use and resistance in animals. And happy to answer questions later. Thank you.
Thank you very much, Dr. Shively. Appreciate the update from APHIS. And as I said, we'll go through some questions here after our remaining two speakers. Next, we're going to hear from Dr. Megan Nichols. Megan serves as the Deputy Director for the Division of Foodborne, Waterborne, and Environmental Diseases at the Center for Disease Control and Prevention. In this role, she works on a variety of One Health topics related to enteric, waterborne, and fungal diseases. She previously worked as the enteric zoonosis activity lead working on multi-state outbreaks of Salmonella and E. coli resulting from exposure to animals and pet products. She is focused her work on investigating multi-state outbreaks of human illness linked to petting zoos, small turtles, livestock with strains of multi-drug resistant Salmonella, and the pet food products. Prior to joining CDC, Megan worked as the principal investigator of the active bacterial core surveillance program at the New Mexico Department of Health for 5 years. With that, I'm going to turn it over to you, Dr. Nichols, for your presentation from a CDC perspective.
Excellent. Thank you so much. And good morning, everyone. So I'm here to talk a little bit about the human angle of One Health and some of the lessons that we've learned from human illness outbreaks linked to animal contact. Now, these outbreaks are unique and give us a different type of insight into transmission dynamics of certain enteric pathogens. And when I say enteric, I'm talking about those, and I know it's a little early, but it's still before lunch, those that usually cause diarrheal illness, but in some circumstances can cause more severe illness. And they're unique because we don't necessarily have a foodborne transmission pathway, but rather a person who's coming into direct contact with animals or the animal environment. Next slide. So when I say outbreak, what exactly is an outbreak? Well, we at CDC use the definition that's been established by the Council of State and Territorial Epidemiologists, or CSTE. And that is when 2 or more people get the same illness and the investigation shows it came from the same contaminated food or drink or contact with the same species of animal. Now, when I say same illness, what I'm talking about there is the same on a molecular level, meaning that we've done DNA fingerprinting on the bacteria that came out of the poop of the person and maybe came out of the food, drink, or the feces of an animal. And it's exactly the same or very, very highly related genetically. And we've done the trace back and research to try and find out a little bit more about the common origins. Next slide. So when we are looking at detection of outbreaks, it's actually a really neat, I'll reference NCIS type of model that's evolved over the past 20 or so years, and that is through our PulseNet network. So PulseNet is a network of public health laboratories around the United States that when there is a person who gets sick and the person goes to the doctor and submits a stool sample, or maybe it's a blood sample if they're hospitalized and really sick, and the bacteria such as salmonella is grown out of that and isolated, that salmonella bacteria then is submitted to a public health laboratory where it undergoes DNA sequencing and we have the fingerprint. Now that fingerprint is compared to other DNA fingerprints of bacteria across the country. And when we identify a cluster of DNA fingerprints that are genetically related, almost identically, and it occurs within a certain period of time, say 60 days, then that cluster or grouping of isolates that usually relates to patients is looked at a little bit more closely by a team of epidemiologists. So it's our laboratory team and our epidemiologists working together to then say, did the patients from which these isolates came have any common exposures? And that's how we're able to identify multi-state outbreaks and say, well, this person who resided, say, in Nevada had the same type of exposure to a certain food product or animal as a person, say, in Virginia. And then look at doing traceback and identifying potential origins of outbreaks from there and doing public health intervention to stop the outbreak. Next slide. We know that outbreaks are just the tip of the iceberg. So when we investigate these, we know that for every one person, one patient that has an isolate that's uploaded into the database and allows us to track a potential outbreak, there are about 29 others that also got sick as part of an outbreak, but we didn't detect them. And part of the reason is some people get sick, but they might not need to go to the doctor because their illness is not severe, or they get sick and they go to the doctor, but an isolate or a specimen such as a stool sample isn't collected. And without those two aspects, we will never know about whether or not those people are truly part of an outbreak. Next slide, thank you. Now, this is especially important in terms of outbreak detection because we know that there are certain groups that are at higher risk from enteric zoonoses. So again, these are mostly some of bacterial pathogens that cause diarrheal illness and are transmitted between people and animals. And we know that there's a higher risk for infection among young children, and that's usually those who are less than 5 years of age. And there's a variety of reasons for that. Chief among them is that young children may be less likely to practice really thorough hand washing, and they might have types of interactions that are definitely more interactive. So if you look at the height of, say, a goat at a petting zoo and the height of a small child, they're nearly identical, which means that a child might have more opportunity to come into contact between their mouth and the railing or be more likely to hug or kiss the animal. We also know that there are certain populations that when infected with pathogens like salmonella, they're at higher risk for adverse outcomes. And that includes older adults, young children, and people with weakened immune systems. So among these groups, if they get sick, they might be more likely to be hospitalized and or have an adverse outcome such as death. So in looking at these outbreaks, we are very motivated to try and to detect them and intervene or respond early, because we don't want there to be these really severe chances of outcomes for these children and others. Next slide. Now, we also tend to see an interesting pattern when we're looking across these clusters and outbreaks that sometimes give us a clue, because we don't know right away how the genetically related group of isolates relates to each other. So oftentimes we're looking for those clues. And one thing we might see, and we saw this in a Campylobacter outbreak that occurred a couple of years ago and was linked to pet store puppies, is we see groups of people that have specific professions or occupations that might be associated with greater animal contact. And so it's not uncommon in animal contact related outbreaks for us to see the occupations that are listed here on the slide commonly pop up as people who get sick in enteric zoonotic outbreaks. Next slide. So what have we learned through investigation of outbreaks linked to livestock specifically? Well, here I'd like to highlight one outbreak that occurred several years ago. And this is an outbreak of multi-drug resistant Salmonella Heidelberg. So what we were seeing is that there was a new strain of Salmonella that was causing severe illness in both dairy calves and in children in a cluster of Midwest states. And that was pretty unusual, because most of our outbreak investigations, although we see people that get sick, it is uncommon for us to see animals that are also getting sick. But in this instance, it took a really astute veterinary laboratory diagnostician to say, wow, I'm seeing a lot of calves that are coming through for necropsy, and they have invasive Salmonella infection. So it's not scours that really cause them to die, but rather the Salmonella is causing them to develop a severe infection that's going throughout the body system. And I'm hearing in this community that there are also people who are getting sick, whether they're those that work with the calves or children who are, say, interacting with the calves via show activities or chores at home. So once this outbreak was detected and we looked at the isolates, one of the things our laboratory saw is that there were cases across multiple states from this same strain, and there was a brand new gene that we had not seen before that was resulting in antibiotic resistance and really severe multi-drug resistance. So resistance to several antibiotics that would be used to treat this infection in people and in calves. So the scary thing for us is that within the veterinary community, there was not an antibiotic that veterinarians could reach for that would actually address this type of infection in the calves, this multi-drug resistant Salmonella Heidelberg, because it was so resistant. And there were some options for human medicine, but they were antibiotics that you usually reached to as more of a last resort. So in order to respond to this, public health, animal health, environmental health, and agriculture experts really work together to track the spread of the strain in both people and animals, and to provide education about how to potentially prevent infection and implement best practices, whether it was cleaning at a livestock market or whether it was talking to groups of children in FFA and 4-H about safe animal handling practices, because we want children to continue to have these opportunities to engage with animals, but do so in a way that doesn't result in them getting sick. Next slide. So what about beyond the outbreak? So we investigate this outbreak and we spread prevention measures, we do some interventions, and we start to see that the outbreak cases, or the number of people who get sick, declines. But that's never the end of the story, because we still have a multi-drug resistant strain of Salmonella that's infecting humans and dairy calves. And so CDC supported the Ohio State University and University of Wisconsin to study some of the contamination in dairy calf production to see if we could work upstream on some prevention efforts and learn a little bit more about how this strain of Salmonella is in the environment. And so you can see here that there were some key findings regarding where Salmonella was found in the environment and how we might be able to address some of those with really good infection control. And that really includes cleaning and sanitation in the environment. Next slide. We also recognized that there was a spot of blindness in what we were looking at, where we got a lot of questions during the outbreak about how did this multi-drug resistant, or this gene that conferred multi-drug resistance that was brand new, evolve in this outbreak? Was it related to, say, another strain that somehow got into the calf production? Was it because there were antibiotics that were putting selection pressure on the bacteria to basically adapt and evolve so that this gene resulted? And we didn't know. And so we also worked on funding a survey of production animal veterinarians' prescription practices and some of the factors that were influencing antimicrobial drug use and perceptions to try and identify on a broader scale, how could we potentially understand what some of the factors were that were driving antimicrobial drug use, and how could we work to actually address them? And I really appreciate that the AVMA has put out a definition of stewardship, because that's one thing that also came out of this paper, is really having kind of a baseline understanding of what is antimicrobial stewardship. If we say we're going to be good stewards of antimicrobials or antibiotics, what exactly does that mean? So I've listed that here on the slide. Next slide. And then next, what can we at CDC do around antimicrobial stewardship? So this is one component of a One Health page, because we recognize that oftentimes in putting out some of the information, we focus mostly on the human healthcare sector because that is part of our public health mission. But we also want to provide information for those who might come to the site about actions that veterinarians are taking to really reduce antimicrobial resistance and provide antimicrobial stewardship in veterinary medicine. And so we have some of the best practices that are listed here on the slide, and this also appears on our CDC website. Next slide. So let's talk a little bit more about antibiotic stewardship education. So we found out a little bit more about this bacteria in the environment, and we found out about some of the drivers around making decisions around antimicrobial use. But what about calf producers specifically, because this multi-drug resistant outbreak was detected there? Well, we also worked with the Ohio State University to develop and test some antibiotic use decision-making tools and a training program for calf producers to see if we could actually change some of the antibiotic use behaviors over time. And after this intervention, one of the things we noted is that there was some improvement in test scores around antibiotic use knowledge. Now, additional time is needed. This was done over a short period of time, as with any pilot project, but we were optimistic that some of the early indications indicated that providing some of these tools did have an impact on antibiotic use and stewardship. Next slide. So I'd like to share with you a video that highlights some of this work. So Morgan, I'll turn it over to you to go ahead and play the video.
My role at Ohio State University is both research and teaching. Ultimately, how we use antibiotics in calves has an impact on human health because it's all One Health.
Antibiotic resistance is a One Health issue. It is important for both animal and human health. And with this issue, we are wanting and we are trying to make sure that each antibiotic that we use on the farm is used responsibly and when it is necessary.
The CDC has long been aware of the issues of antibiotic stewardship in human health and veterinary medicine. They recognized a need for antibiotic use training materials for calf producers.
We started out with a small group of farms to actually make sure that this training was indeed effective. We worked with the veterinarian and the calf producers side by side, teaching them, how to identify sick calves versus healthy calves and when antibiotic treatment might be necessary.
As a clinical veterinarian, I'm always trying to weigh proper antibiotic use with the proper and humane treatment of the animal. Working with the team at Ohio State University has been great. The more we can educate people, both the public and the producers, about how and why we use antibiotics, the better off we're all going to be.
As a producer, I know how important it is for animal welfare and human safety in this industry. Since working with Dr. Habing's team, we've really focused on using antibiotics when we really need them. And what we've seen is that when we're saving the antibiotics for when we really need them, they've been working a lot better.
Health and welfare is a shared and common goal between calf producers, between scientists, between consumers and veterinarians. So really this shared goal brings all of us together to create research and design programs like this intervention that just formed a really natural partnership that we then continue to have with the CDC.
Thank you. So one of the things I just want to highlight is I think at CDC, we recognize that there are things that start upstream, that downstream might impact human health. And without really looking at this from a One Health perspective or approach, we're not going to be able to solve some of these really, really challenging issues. But also in order to do that, it's really creating space for those with expertise in the environmental health, in agriculture, and in veterinary medicine to come forward with their expertise and work together. And so we were really happy to provide some pilot funding in order to help that engagement take place. And are really excited to see where that work goes in the future. Now, I also wanted to highlight some recent activities at CDC, because I think one of the things to look at is where are we going next? What is next? So opportunities for collaboration, but also wanted to highlight some of the recent reports from CDC, especially the one from this year, looking at the impact of COVID-19 on antimicrobial resistance. And using that, how can we potentially look at some of the gaps, look at some of the opportunities for investing in our public health infrastructure in a One Health approach to look at the challenge of antimicrobial resistance and really create some great prevention measures to improve stewardship. Next slide. I also wanted to highlight, and you saw on the last slide, we have the 2019 threats report on antibiotic resistance in the United States, and that focuses primarily on some of the human and also One Health aspects. But there is a report that the AVMA did looking at animal health, and so I wanted to highlight this because I think it's really, really great to see this resource that talks a little bit more about antimicrobial resistant pathogens affecting animal health in the United States. Next slide. And Morgan, I'll have you go ahead and click through these, but there's some really great resources that are included in that, including multiple different species. So dogs, cats, cattle, chickens, turkeys, there's some on horses and other areas that really identify some pathogens of concern, what we need to know about that, and then actions that veterinarians can take to address these. I also didn't have awareness of a lot of the challenges that the fish and shrimp groups face when it comes to antimicrobial use and stewardship. So this was a very interesting read for me as well. Next slide. So as others mentioned, I think one of the areas we're also going next is to look at the whole environment of people and animals. So not only looking at the livestock aspects, but looking also at the animals that we share our home environment most closely with, and some of the antibiotic resistance that we do see in pets and people, and how do we help to change some of the practices there so that when antibiotics are used, they're used in a way that is really promoting stewardship and some of the best practices as outlined by AVMA, and ensuring that we maintain the effectiveness of antibiotics for humans, animals, and the environment so that they're used when needed, and when they are, they're effective. Next slide. So if you want to learn more about any of the enteric zoonotic outbreaks and or antimicrobial resistance, I put some links on the slide and a webpage that highlights some recent activities by the team. So with that, I'll turn it back over to you. Thank you.
Thank you very much for your insights, Dr. Nichols. And again, we will have Dr. Nichols joining us for the question and answer discussion period after our final speaker. A reminder that if you have questions that you would like to pose, please put those in the question and answer box that's at the bottom of your screen. We would prefer to not get those in the chat, so use the question and answer. And you can go ahead and put questions from earlier speakers in there while our final speaker is presenting. With that, our final speaker before we go to the question and answer session is Dr. Allison Robertson. Allison's a professor and extension plant pathologist at Iowa State University. Through her extension program, she strives to increase the plant pathology expertise of her clientele to facilitate more effective disease management and increase net returns for farmers. Her research program complements her extension by addressing current issues in disease management of corn and soybean production. For the past 15 years, she has evaluated fungicide products as seed and foliar applicants for the management of various diseases. And she's been here at Iowa State University since 2004. Welcome, Allison, and we appreciate your giving us some insights on this panel this morning from the crop perspective. Please take over.
Thank you, Paul, and thank you to the Farm Foundation for having me here today. So can we have the next slide, please? So how many of you know that plants get sick? This had never occurred to me until I had a 1-week class in plant pathology during my freshman year at college, and I was totally fascinated and fell in love with plant pathology. So in the short time that I have today, I'm going to briefly review plant disease or crops. We're mainly talking about crops here, and their management, in particularly focusing our efforts on how to steward fungicides, or what many of you would refer to as antifungals. Next slide, please. So crop diseases are very important. They reduce yield, they might impact crop quality, and they also reduce profits and income for farmers. Next slide. So like animal and human diseases, plant diseases can be caused by fungi, bacteria, viruses, and then also we have nematodes that cause various crop diseases. Next slide, please. We manage or treat crop diseases using a variety of tools, just like you do in animal and human health. Next slide. So these tools can include planting disease-resistant cultivars, and this is always the cornerstone of our disease management, or of, I say disease management, you might say treat plant diseases, but we're always focused on planting a disease-resistant cultivar. And so we work very close with crop breeders to breed these disease-resistant varieties. And then we might also apply fungicide applications to the seeds of the crops, and we can also apply those foliarly. So a fungicide would be what you would call an antifungal. And then thirdly, we'll use cultural methods such as crop rotation. So rotating corn and soybeans and wheat and alfalfa in terms of field crops. We might adjust planting dates so that we can avoid different diseases. Tillage might be used because many of the pathogens that we're talking about survive in the crop residue, removing the infected crop debris. So there's a lot of different cultural methods that we can use. Next slide, please. So in the light of why we're here today, I'm going to focus on the use of fungicides or antifungals to treat or manage crop disease. So the Fungicide Resistance Action Committee is an international committee that classifies fungicides based on their mode of action. And there are currently 45 FRAC groups. Of those FRAC groups, we use 3 to 7 of them on field crops. And I'm going to focus a lot on field crops just because that's what I'm more familiar with. Next slide, please. So among those 3 to 7 FRAC groups that we use, there are 3 groups that we use primarily. And those are FRAC group 3, which are the demethylation inhibitors, or the DMIs. The FRAC group 7, which are the succinate dehydrogenase inhibitors, or SDHIs, and then the FRAC group 11 quinone outside inhibitors, or sometimes we refer to those as strobilurins. Next slide, please. So what's important here is that our DMIs, our FRAC group Is, are essentially the same as the azoles that are used to treat fungal diseases in humans. Next slide, please. So that comes to why am I here today? The reason why I'm here today is because there's a growing concern that the use of DMIs, the fungicides, the FRAC group 3s that we're using in agriculture is resulting in azole resistance in human health. And as you might imagine, there are research papers that support this concern, but there are also research papers disputing this concern. Next slide, please. So I'd like to step back a minute and just talk about field crop production in the US, in particular corn, soybean, and wheat. And you can see this on this slide that these crops occupy approximately 2.5 million acres in the US, and that they're very, very important to the US economy. And the thing about these crops, though, is that they are threatened by disease. So next slide, please. So if we take corn as an example, this slide shows you some of the most important diseases that we have on corn. We have tar spot, which is a new disease that was first reported in Indiana in 2017 and has now spread throughout the US. And then we also have the other leaf spots that have been around for a fairly long time, grey leaf spot, northern corn leaf blight, and southern rust. But when we look at the losses due to those diseases over the past 10 years for grey, northern, and southern, and then over the past 5 years for tar spot, you can see that losses can vary from tens to hundreds of millions of bushels each year, or from a few cents per acre to over $20 per acre. And one thing to remember about field crops is that they're not high value crops like tomatoes or grapes. So farmers are not making a lot of money on these crops. And then the other thing to remember about managing these diseases or treating these diseases with a fungicide or an antifungal is that costs the farmer approximately $10 to $30 per acre, depending on the product that he uses and the method of application that he uses. Next slide, please. So we use treatments with antifungals to mitigate those losses due to disease. Next slide, please. Okay, so in the agricultural community, we're aware of fungicide resistance developing among our plant pathogens. And in fact, the Fungicide Resistance Action Committee monitor important plant pathogens around the world for their sensitivity to fungicides that are being used to mitigate disease. So for example, over 40 species of plant pathogens have been reported to be resistant to the group 11, the QOI fungicides. And in the US, the frog-eye leaf spot pathogen that you see here on soybeans, there's a picture there, is resistant to this group 11 fungicide. And so consequently, we rely on the group 3, the DMI or the azoles, and also the group 7 fungicides to mitigate disease on soybean and protect yield. Next slide, please. So communication and education is key to protecting the antifungal fungicide tools that we have in our toolbox. And so to this end, we've been doing a lot of communication and education. This is an example of some publications that are available to farmers to enable them to choose the best antifungal in terms of efficacy to treat their crops. And so this is available from the Crop Protection Network, which is a website where all the field crop pathologists work together to provide educational materials for farmers across the US. In addition, in our professional society, the American Phytopathological Society, at the 2021 Plant Health meeting, we had a keynote address from Dr. Maren Brewer from University of Georgia, she spoke to us about does the agricultural use of triazole fungicides contribute to antifungal resistance of Aspergillus fumigatus in humans? And following on from Dr. Brewer's address, us field crop pathologists who are part of the Crop Protection Network then got together and co-wrote an article addressing this same issue. So this slide shows that in addition to communication and education, research continues to enable the stewardship of antifungals in agriculture. And so a lot of this includes improving and developing new models to predict crop disease and enable farmers to more judiciously apply fungicides to their crops. So an example here is a web-based tool, which is the Fusarium Head Blight Prediction Center for Wheat. This web-based tool has been available for the last 20 years, and it gives farmers a real-time prediction risk of whether head scab is going to, whether their crop is at risk for head scab and whether they should be applying a fungicide or not. More recently, Dr. Damon Smith at the University of Wisconsin has developed a couple of smartphone apps. So this is Tar Spotter for tar spot on corn, and then Sporecaster for white mold on soybeans. And both of these are based on models of how the disease develops. And so these are tools that a farmer can use to determine the risk of that disease developing in their field and whether they need to apply a fungicide or not. And most recently, within the last 3 years, the National Prediction Modeling Tool Initiative was initiated. And this initiative is working together on wheat and corn and cotton. And we are developing models about diseases that are occurring on those plants with the view to developing tools that farmers can then use to apply fungicides more judiciously to manage those diseases. Next slide. So I'd like to thank you for your attention. Thanks to the Farm Foundation again, and I look forward to our discussion.
Thank you very much, Dr. Robertson, for that presentation on crops. And now I'm going to ask all of our speakers to join me back here on the panel, and we will start going through some questions. I would like to remind the audience that you can submit questions as well using the question and answer function at the bottom of the screen. And we will get through those questions as time permits. While we have some already, but while more folks are putting some in, I will lead off some question. And just interested, certainly from my experience working with Farm Foundation and previous Farm Foundation webinars, certainly I think the audience has a lot of interest in food security issues and how we assure food security as a growing world population. And so I'd like to throw that out to the panelists to make any comments that you would have on the importance or the role of judicious antimicrobial use in livestock and crops and the role that plays potentially in food security. So open it up for panel discussion if anybody has comments.
I think I'm happy to start, Paul, because I think I probably have an easier answer than some of the rest of our panelists. But in terms of looking at food security around the United States, one of the things that we recognize as a critical component of that is our ability to produce safe food. And alongside that is really making sure that the animals are produced in a way that allows them to be healthy. So I would say that looking at antimicrobial resistance, that can certainly pose a risk to animal health and human health. And so having access to antimicrobials and making sure that they're still effective is absolutely critical to maintaining animal health and subsequently to having safe food and ensuring food security.
Thank you, Megan. Dr. Shively, it looks like you were getting ready to perhaps unmute. So.
I don't know that I can add a whole lot more, but I will just say from an animal health perspective, it is really important that we preserve the effectiveness of antimicrobials for addressing our animal diseases too. So again, aligning with what Dr. Nichols has already stated, we must use antimicrobials judiciously in order to protect both human and animal health and ultimately food security. Thanks.
Excellent. Anything to add, Dr. Robertson or Dr. Flynn?
I was going to say that animals and humans rely on crops. That's where we start off, right? We're the bottom of the pyramid, and unfortunately, crops do get diseases, and antimicrobials are very important in that world to manage those diseases. And but we also, yes, we do have this resistance problem that threatens the profitable production of those crops. And so how can we steward those antimicrobials better so that we can ensure that we have food not only for us but also for our animals?
All right, thank you. So now I'm going to ask an audience question, and this is focused for you, Dr. Flynn. On the slide, looking at the trend of resistance declining in chickens since 2017, one drug, the questioner thought it was maybe the green line, showed a significant increase. Have you determined what might have occurred and if that's a true increase in resistance? I think he's maybe looking at the slide to see what the question is there.
Yeah, sorry. Yeah, it was a lot of information presented on those slides, and I think we have to step back and sort of look at kind of the trends holistically, and be careful as far as I think we do see in certain cases, trends that may trend up and down, from year to year. And even looking at the slides I presented is a fairly limited sort of snapshot in time, right? So again, I would sort of put that sort of qualifier around it as far as just how much we can conclude from, any one dip or rise on any given year. And I think that might have been, they may have been referring to one where we saw a tick up for 1 year. But again, I think we just have to be careful how much we can interpret, those sort of short-term shifts, which is the challenge with monitoring AMR, is needing to have a sufficient database and to be able to look at a sufficient period of time to draw conclusions.
Excellent, appreciate that. So I'm going to jump around the questions here a little bit, and encourage the audience to continue to add questions. But this one is broadly focused towards all of the speakers, perhaps Dr. Robertson, a bit more towards the federal government folks, because the question is, what is the government doing to incentivize startups innovating in the AMR space? There are many programs in human health, but what about on the agricultural side? So.
I can jump in from the USDA perspective. Our Agricultural Research Service has a research program that is focused on looking at alternatives to antimicrobials. But I will say that this is a gap in our current activities to address AMR. Thank you.
Yeah, and adding to that, I mean, certainly continuing to have access, ensuring that veterinarians, farmers, ranchers continue to have access to effective therapies for managing animal health issues, is critical. And, which is one of the challenges with AMR is certainly efforts to foster or encourage judicious and more limited use means that there are these pressures to reduce a volume of use of these products, which can be, a disincentive for, companies in the animal pharmaceutical, and this is certainly an issue too on the human healthcare side, a disincentive for companies to develop those products because inherently there's a desire to sort of limit their use, to curb resistance. So it is a real economic sort of challenge in terms of and a gap that still needs to be, I think we need to continue to focus on it because we need to make sure that there's a continued pipeline of products and the animal pharmaceutical industry overall, is a very small sort of industry. So I think that's going to continue to be an issue. I think we also, as Chelsea said, I think are looking towards, what we can do to support development of alternatives, well, not only alternative therapies, but also things that can be done in the animal health sector to sort of reduce the need for antimicrobial therapies.
Absolutely. And in response to this, I also put a link to some of our, it says, I don't know who asked this question, but appreciate it because we have a webpage for CDC called Innovative Projects to Combat Resistance. And so in terms of this, one of the things I do want to highlight is at CDC, one of the main drivers is looking at the data. So data-driven needs, essentially, and that might be through data collected during outbreaks or through some of our routine surveillance. And when we identify those, it's then looking at how can we help fund some of our partners who have expertise or innovative ideas in that space. And we do that mostly through BAAs or broad agency announcements. So there's a link to the webpage that shows a lot of those innovative projects that have received funding, and this is across the One Health spectrum. On the animal health side, we have some CDC designated food safety centers of excellence. And so these are typically collaborations between health departments and academic centers that have veterinary schools or really robust animal health program to really look at how do we create space for some innovation and provide some seed funding, often for pilot projects in that area. And it's been really great to be a part of that. I think it's really helped us to look beyond some of our narrow scope to think very broadly about how that collaboration can further some of these efforts and further these needs, and recognizing that many of those partners have ties into the animal producer community that we at CDC or even in the government don't have. And so an ability to hear from them directly about what they're seeing and what some of their needs are. So I put that in the Q&A and hope you have a chance to peruse.
Great, appreciate that. Now a question for Dr. Shively. Does the Noms, National Animal Health Monitoring System, look for alternatives to feedlots or look at alternatives to feedlots such as pasture-based livestock management as a method to reduce antimicrobial use?
Yeah, thank you for that question. And I just want to reiterate something that Dr. Flynn just mentioned, which is that overall, we are not focused on reducing antimicrobial use. We are trying to find ways to reduce the need to use antimicrobials. And that can be through a variety of different practices, so preventing disease on farm through vaccination programs and improved biosecurity, or finding alternatives to antimicrobials. So I just want to start with that, we're not trying to compare different animal management systems against each other. We're just trying to better understand the issue and find ways to reduce overall animal disease issues on farm. Second, I just want to mention that almost all of our beef cattle do spend at least part of their life on grass, and Noms does conduct studies of our cow-calf segment of the industry, as well as the feedlot studies that I mentioned. And so for those of you who are familiar with the cattle industry, you'll recognize that this leaves a gap of our stocker or backgrounder segment of the industry. And we recognize that this is a very important phase of a beef animal's life cycle, but it is a difficult group to survey. It's very fluid. The producers that are involved in that practice, it changes depending on the price of corn, the price of feed, the price of of beef. And so trying to get a list of those producers that we could go back to year after year to survey has been really challenging. So if folks have ideas on how we could survey, I know that that has been an interest at Noms for a while, just to better understand that stalker or backgrounder segment of the industry. But yeah, so we try to look at the phases that are relatively consistent, so the cow-calf segment and the feedlot segment of the industries. And then I'll just remind everyone that the dairy industry is also ultimately a part of the beef industry as well, and Noms does conduct surveys of the dairy industry. So we're trying to capture as much data as possible and trying to understand where the majority of these animals spend their life cycle. Our Noms studies, we try to represent at least 70% of the animals and 70% of the operations that make up that industry, so that we can provide information that's useful to a broad segment of that industry. Thanks.
Great. Thank you, Dr. Shively. The next question, I think applies to all, and Dr. Robertson, there's a specific call out to you for that. So, and this is a question broadly related to, are there certain diseases that each of you see in your different monitoring efforts that are particularly prone to antimicrobial resistance, and specifically, Dr. Robertson, you know, on the fungicide perspective, has your program's website identified any specific fungicides that are particularly problematic, and how closely do you work with CDC or other entities on those?
Thank you for the question. Thanks, Paul. I was just answering it on the chat, but yeah, so I can talk mostly from the field crops pathologist, but we do monitor the pathogens that we have on corn and soybeans and wheat annually for their sensitivity, primarily to the FRAC groups 3, 7, and 11. So although we have 20, 30 products available to be applied to these field crops, those products have different active ingredients that just belong to these 3 main groups. So we do see slight differences between the active ingredients, but essentially sensitivity to FRAC group 11 is a very big problem, and so that's why we're very reliant on that FRAC group 3, which is the DMIs or the azoles, because we're just losing this efficacy with the FRAC group 11. The other thing that I was going to point out is that sensitivity to these different FRAC groups will vary among different species. So, for example, the frog eye leaf spot pathogen on soybeans is caused by Cercospora sagina, and that's a one mutation, and the fungus is either susceptible or resistant, and that's it. Whereas in the grey leaf spot pathogen, which is also caused by Cercospora, but a different species, Zeomatis, if that mutation occurs in that species, it is, I can't believe I forgot the word, but the fungus is not able to survive. So it's a lethal mutation. So yeah, I mean, it just varies depending on the species, and then depending across the different groups. And no, we do not work closely with the CDC. So I hope I answered your question.
Yes, other comments from the others, if there's particular diseases or organisms. If not, we'll move on to the next question, but happy to have any other comments.
I'll just comment quickly. I think this is a great question and something that we're still learning a lot about. The National Animal Health Laboratory Network AMR pilot project is the first time that we're really looking at a national level of animal pathogens and looking at resistance in them. So, we have 3 years of data that are published in reports, and I believe 4 on our dashboard. So I encourage you to take a look at that. But I do just want to say that one of the challenges there is that we don't always have clinical breakpoints to be able to interpret resistant versus susceptible when looking at specific pathogens and drug combinations in our animal species. And so that can limit our ability to understand, overall resistance, just because we don't have the information needed to make that determination. I will say, just from looking at the latest report from the Noms data, we do see some multi-drug resistant E. coli, particularly in our cats and dogs. So that's something that we are seeing. I think there's a lot of work to be done to understand more about that. And then Mannheimia hemolytica is another one that affects our cattle. And that's one where we are starting to see some resistance. I know that one of the projects that we've been looking at is how can we do better sampling so that we can really understand at the population level what that means. Because the non-AMR pilot project, these are samples that have been submitted to diagnostic labs. So a lot of times these are animals that have been treated. So it's not necessarily representative of the overall animal population, but of the sick animals specifically. So I would just say more to come. Take a look at that non-AMR pilot project for some initial results. And I think there's still a lot of work to better understand this.
All right. Now a question on cooperation across agencies. So the question specifically asks, how closely does APHIS and CVM, FDA, CVM coordinate and share data and evaluations of trends? But I will broaden that question to also, several of you mentioned NARMS and, a collaboration across all three of your agencies. So would open it up to one of you, perhaps Dr. Flynn, to start with, on kind of what types of interaction between the agencies occur and how you share data, and then folks can comment on the collaboration through NARMS or other collaborations as well.
Sure, Paul, I can start. I mean, I think NARMS is a great example. I mean, that's a longstanding collaborative program initiated in 1996 and has been expanded ever since then. Again, it's a collaboration between FDA, CDC, and USDA, and sort of each agency, sort of contributing to the overall program, where FDA, is taking lead on sort of coordinating, collecting and putting into or submitting into the system data that's being collected from retail meats across the U.S. in terms of surveillance or monitoring on foodborne pathogens. CDC contributing data from the human health sector on foodborne pathogens, and USDA FSIS contributing data that they're collecting from animals at slaughter plants. So it gives us a sort of cross-section view across those different sectors, again, with the common thread sort of focusing on monitoring trends in terms of what is happening in foodborne pathogens, sort of recognizing that that's a place where you have movement of pathogens from the food animal sector into the human, potentially into humans, and along with that, the potential that those pathogens may be resistant to different drugs. And again, as we talked earlier, some of those drugs are shared by both human health and animal health. So I think it's a good example of where we've been working collaboratively on collecting data on resistance. And certainly there's other areas that we work very closely with both USDA and CDC on. But anyway, that's certainly one good example.
And I think, as others have alluded to, I think NARMS gives us some really exceptional data. I also want to highlight that one of the things that we have is a really great collaborative working relationship amongst the agencies and others that may not be represented here. So for example, FDA, CVM, and CDC have regular standing meetings where we are talking about some of these emerging challenges or about really specific outbreak investigations. So for example, the pig ears related salmonella outbreak outbreak that occurred a few years ago, CDC recognized that our part was to really look at the data as it related to human illness and to partner very closely with the experts at CVM who had knowledge about how this contamination of a product might potentially have impacts on animal-related illness or animal health. Similarly, with the multi-drug resistant Salmonella Heidelberg outbreak, we worked very closely with USDA APHIS because we recognized that CDC had an interest in the public health items and wanted to run any ideas about future projects or other things by our APHIS colleagues to make sure these made sense. But also during the investigation, APHIS actually had a study that was initiated with the producer community in the affected area to look at on-farm practices, everything from open herds to closed herds to nutrition and various other metrics to try and find out a little bit more about the impacts on animal health and what activities might be needed there. And it evolved into partnerships with ARS and others. So it's really a collaboration that is ongoing and essential in order for us to all really work on this, because if we just looked at the human illnesses, I think we wouldn't even begin to touch the surface of the ways we could potentially learn from and prevent some of these illnesses in the future.
All right, appreciate that, Dr. Nichols. And in just a minute, I'm going to let Dr. Shively answer the question, but in just a minute, Dr. Nichols, I'm going to come back to you. You mentioned the Pig Ears Project, one I'm familiar with, but others on the call may not be as familiar and wondering what in the world you're talking about with pig ears. So I may come back to you for just a quick overview of that particular outbreak. So Dr. Shively, on this question of federal government interaction and relationships.
Yeah, thank you, Dr. Plummer. And I just want to say, I think my colleagues highlighted some great examples of collaboration. I think working from a One Health approach and working together between our agencies is incredibly important. And that's how we're going to be able to move forward on the issue of antimicrobial resistance. I do just want to say from the USDA perspective that data confidentiality and protecting our producers is at the front of our minds when working on AMR and many other issues, because we recognize the sensitivity of this. When we collect samples with one of our Noms studies, for example, the information of who that producer is, where they're located, anything that's identifying, that's never shared. That information is only known by our field veterinarian. Even us and at headquarters don't know that information. So I just want to make sure that our audience understands that while we do a lot of data sharing and try to address outbreaks across our various agencies, we are trying to make sure that we're protecting the identity of all the people that are contributing information into our system.
Wonderful and very important point there, Dr. Shively. I appreciate that. So, Dr. Nichols, back to you for a quick overview of the pig ears and I think a great example of the One Health kind of nature of this issue.
Yeah, happy to expand on that a little bit. We noticed a couple years ago that we were starting to see illnesses of a particular strain of Salmonella. And in looking at the information about the different things people had ate or drank or swam in or potlucks or restaurants, one of the things that came across very clearly was that almost every person in this outbreak reported having a dog. And while that's not uncommon in the US population, the other thing that they were reporting is feeding their dog treats. And looking at our normal population in the United States who doesn't get sick or who's just been surveyed about what they've done in the past, it was only about 16% of people who were healthy, who were surveyed, who said, yes, I've given treats to my pet in the last week. And 60% of those who were sick reported giving treats, and specifically pig ears. So we worked very closely with FDA, CVM, state partners, and even some academic partners to go out and collect pig ears from the homes of people who got sick or from the pet stores where they reported purchasing them. And lo and behold, not only did we find the strain of Salmonella that was starting to infect people in this outbreak, we found a lot of other strains of Salmonella and then started finding additional human illnesses that were related. So we relied very, very much on the expertise of our colleagues at USDA, because a lot of their expertise relates to processing of the pig and what happens to the ears of pigs when they are processed, and then also FDA CVM for the pet treat expertise to find out a little bit more about what we could do to reduce contamination. And I'll say that this is certainly something that we find very valuable, not only with government, but also with academic colleagues, is to say, okay, we have this problem. we know we may not have the expertise in-house. And so who can we bring in, rely upon, consult with to help us better understand this and work together to solve these problems?
And if I'm not mistaken, there was also kind of some impact from importation of some of those. So even our international partners were important in that, if I remember correctly.
Absolutely. We found one particular strain of resistance that we utilize Noms to monitor for different strains of resistance. And this one we actually don't see in the United States. And so looking at a particular strain of Salmonella, one of the serotypes carried a specific resistance gene, which is much more common in one of the countries in South America that we don't see typically in the United States. And then when we looked where these specific pig ears were being imported from, they were coming from that same country. So it helped us also try and track back to what may have been a point of origin for production.
All right, great. Next question, what is the relationship among animal nutrition and gut health research, for example, prebiotics, probiotics, postbiotics, in antimicrobial impacts, disease resistance, and animal health? So kind of relating again to the alternatives to antimicrobials. And so any of you have some comments that you'd like to share on that?
Well, I'll jump in and say this is not my area of expertise, but I will point you again to our Agricultural Research Service because they are looking at alternatives. But within APHIS, this is something that we are looking at from a different angle. So with our more recent Noms surveys, for example, we've been asking questions about what sort of other practices are you using on farm? So that might be prebiotics, probiotics, some of these other newer products to better understand the uptake and utilization across our various ag commodity groups to, to see who's using what out there. And we have one project right now with a collaborator where we're taking fecal samples that were collected in our Noms Dairy 2014 study from dairy heifer calves and looking more closely at it. So looking at what we would call the fecal microbiome and the resistome. So trying to understand what impact having different exposures to antimicrobials on these young dairy heifer calves had on their gut microbiome. So specifically looking at medicated milk replacer, as this was back in 2014, so a little bit different now, and also any sort of treatments that they had, because we have all of the diet and treatment records for those calves during that pre-weaning period. So that's just one example of a project where we are looking at this. I don't have any answers for you yet, but I do think this is an area of a lot of interest in trying to better understand the overall microbiome and what's going on and how antibiotics interact with that and impact the overall gut health.
And maybe just adding to that is, and back to the theme, I think this falls in the category of needing to, or additional emphasis on looking at alternatives. And, as there are efforts to sort of look for ways to reduce reliance on sort of traditional antimicrobials and looking for alternative therapies and alternate methodologies to manage disease in animals that, again, at FDA, I mean, we're looking at sort of how we look at these different products in terms of things we may regulate as animal drugs versus things we may regulate as feed additives, which are also other tools that producers may use. So that is something we're looking at in terms of sort of revisiting how we look at certain products, and with the idea in mind that there's additional interest in looking at new technologies for addressing animal health issues.
All right. Next question we're going to cover here is, I believe, a bit focused on you, Dr. Flynn. So as we approach the timeline in June 2023 with the over-the-counter sales of all dosage forms of medically important antimicrobials will be ended, as you discussed in your presentation, what are some of the greatest challenges that FDA anticipates with this change in antimicrobial use policy?
No, thanks for that. I think the what we have been focusing on is sort of recognizing that there are farmers and ranchers that have relied on these products in terms of addressing animal health needs on their farms, and so it is an issue, I think, of access, right? Making sure that there's as these changes occur and as we approach that timeline, that they have the information they need to sort of understand how they can ensure that they continue to have access to products. I think in depending on where you are in certain locales across the country, access to veterinary services may be limited, which is also a challenge in terms of just being able to physically access veterinarians if they're in more remote areas. And so that can be a challenge to them. So I think that's one of the important challenges is making sure that farmers and ranchers can still access the products they need. And I think that is something we're focused on and continue to emphasize that as we move towards, approach the June 2023 timeline, and certainly making sure they understand too that transitioning these products to prescription status, does not necessarily mean that they need to have a veterinarian visit their facility every time an animal needs to be treated, that they can establish a relationship with a veterinarian and work with that veterinarian to make sure that they have products on hand in anticipation of what the issues that they may encounter on their particular farm. So it does certainly it's an added burden in terms of planning around those kinds of issues. But so anyway, I see that as one of the challenges is addressing those situations where folks have been relying on those products.
Wonderful. I think we have time for perhaps one more question for the group as a whole. And so I'm going to take one of the questions that's here and then perhaps broaden it slightly. But the question is stated is, with the increased research and development and then subsequent commercialization of cell-cultured proteins or lab-grown meats, what research or insights do we have on antimicrobial resistance and use within use or emergence of resistance within these settings and processes? And I would broaden that to also to even some of our growth of vegetables or crops, aeroponics, the crop side, particularly the kind of crop growth of leafy greens in different environments. Any comments on risk of antimicrobial resistance there? Do we see antimicrobial resistance, or do these offer a completely safe alternative?
I guess I can start. I mean, I would say that they certainly, these kinds of new technologies certainly provide promise of again, strategies for being able to alternative methodologies for producing food or for managing and trying to address concerns about antimicrobial use and antimicrobial resistance. I think, though, whether it's maybe going a bit far to say that resolves all risk, I think it certainly we need to be cognizant of sort of what what the potential issues that may arise as we and I think that applies to any new technologies as we explore them, that we have to also be thinking about what the potential implications are and being able to manage those risks even for the new technologies. So again, I think some of these have promise, but they also certainly create perhaps different sets of challenges for the ag sector as well.
I agree with the assertion of my colleagues that there's no way to mitigate all risk, and so it doesn't inherently make something safe. But what I think is oftentimes when we look at these different innovative ways of producing different products, it's good to look at the same things that we look at when it comes to traditional production, and that includes sanitation. the environment? Are there any antimicrobials, antibiotics, antifungals that are used in the production to make sure that there's not introduction of contamination into these environments? And so I'm thinking about use of, say, antibiotics in cell cultures to prevent introduction of contamination. And we have seen some of this kind of come to fruition. We did have an outbreak recently that was associated with some indoor grown leafy greens, and an outbreak that subsequently resulted there. So I think that as we expand into new different realms of production and look at the potential for these different innovative methods to potentially contribute to safety, we still need to look at a lot of the other aspects and evaluate those on these individual scenarios to see what impact they may have. And along with everything else, I think it's certainly a balance of making sure that the environmental sanitation process, especially as it relates to food, is really robust in these settings.
Well, my colleagues have done a great job of answering the question, and I have nothing more to add on that. I will just say that one of the things that I think is great about the U.S. food supply chain is that we have choice. It's great that there is innovation and that there are new technologies so that we can continue to feed a growing population and try to do so in a safe and healthy and effective manner. So yeah, I just want to say that it'll be exciting to see all that is to come with cell cultured meat and other new technologies as they come on board.
Allison, did you have a comment?
No, I have nothing to add. I think that Megan in particular did a great job in pointing out things that exactly I was thinking about. So thank you, Megan.
All right. Well, I appreciate that feedback. And as was mentioned, we had some questions about innovation and continuing to develop innovation here. And as we have those new innovations come along, it's our role in society and as researchers and as agencies to kind of continue to understand how that changes the landscape, and appreciate the feedback and input of everybody in that process. And even as we think to some of these new innovations, oversight and kind of approval of alternatives and all of those types of things for these non-traditional type of products come to light and continue to challenge us with new ways to get approvals and review of those. I want to finish up this presentation by thanking our 4 panelists. We appreciate you for joining us over these last 2 hours. I've found the conversation and the questions and your presentations insightful and interesting, and appreciate each of you taking time from your busy schedules to join us here. We'd also like to thank the audience for joining us as well and providing these great questions. Unfortunately, we didn't have time to get to all of them, but appreciate you putting those in and building on the discussion as we have been able to have that. And with that, I'd like to turn it back over to Martha for closing us out.
Thank you, Dr. Plummer, and thanks to all of our speakers for the insightful discussion today. I'd also like to thank all of you in the audience again for the questions. We appreciate your engagement with us. We'd love to hear your feedback, so please take a moment to share your comments about today's session in the brief survey you will see at the conclusion of this forum. If you'd like to help us continue providing valuable programming such as today's forum, I'll quickly remind you about becoming a friend of Farm Foundation. Check the link provided in the chat to learn more. Each and every gift to Farm Foundation strengthens our ability to address rapidly evolving issues impacting agriculture, the food system, and rural communities, and we are so grateful for your support. Thank you once again for joining us today, and we look forward to seeing you at future events.